Okay, we have seen last week basically how proteins and enzyme works by folding their substrate, by arranging the atoms within the substrate to a certain configuration so that they are at the right position to react. We have seen that proteins need, enzymes need something else very often than just amino acid. Like for example, these cofactors, like we have hemoglobin here, and heme from hemoglobin, and we have retinol from photoreceptor there on the left. The next part of this lecture is how do you regulate these enzymes? You imagine the cell has the whole genome, and enzymes are encoded in that genome. And a cell wants to be able to regulate at any time the activity of enzymes. So can you maybe tell me 3 points, three levels on which the cell can act to regulate a reaction that is mediated, that is catalyzed by an enzyme? Can you give me 3? 3 levels on which the cell could act. It's exactly the same as if I was asking you. Let's say you are boss of a company and you are making shoes. And how can you produce more shoes? That's exactly the same question. That's exactly the same question. You want to produce more or less? I mean, which level can you act on if you are the cell? You have a reaction, a given reaction that is catalyzed by a given enzyme. How can you increase or decrease this reaction? What are the levels you can act on? Think about all the cell, what the cell contains. What do you think? I think the air at 24 degrees, so I will be cold and motivated. So, what can you do? What can you do if you want to change how fast or how efficient a given reaction catalyzed by an enzyme is?
Increasing the amount of it?
Yeah, that's the first thing, right? That's the first thing. You just increase the amount of one enzyme. You have this enzyme that mediates that reaction. You just increase the amount of the enzyme. So the reaction will be boosted by the increase of enzyme. That's the first thing. You can increase the amount of enzyme. That's one. There are 2 left.
Increase the amount of substrate.
You can increase the amount of substrate. That's a good point. But unfortunately, you can increase the amount of substrate up to a point at which the enzyme will be saturated. So sometimes increasing the amount of substrate, if you do not increase the amount of enzyme, will not be so much efficient. But that's another way. Yes.
So we have to make them move faster, so there's more
Yeah. So that's very good. So the first thing you said, so you can increase the amount of enzyme. That's great. I'm saying also you can also decrease the amount of an enzyme. You increase the amount of an enzyme, for example, by promoting transcription of the gene that codes for that enzyme. You can decrease the amount of an enzyme by maybe accelerating its decay and breaking the enzyme. Okay. But then there is another point which you just mentioned, is you don't change the number of enzymes. So you have 1,000 copies of that enzyme. You want to keep the same number, but you are just regulating the activity. What is the difference between changing the number of enzymes, either decreasing if you want to decrease the reaction, increasing the expression if you want to increase the reaction, or changing the activity of the enzyme? What are the benefits of each regulatory level? What do you think is the advantage of regulating the number of the enzyme? What is the bad point of regulating the number? What is the advantage of just changing the activity? What are the pros and cons of each regulation?
So like, if they move faster and stuff, then it creates more entropy. Increase entropy, so it produces more heat. No?
Yeah, that's all right. That's all right. That's all right. That's not a problem. That's not a problem. All in all, it will not be much. Let's say you want to do exercise, and then you need to sort of activate the reactions of all your muscles and everything. Then you will generate more entropy. You will release a lot of heat. That's okay. That's okay. That's not a problem. But yeah, you are hinting on something which is the efficiency, which is a bit linked to what I want you guys to tell me.
Well, to expand on Riku's one, if you have more energy, then your reactions occur faster, so then the next substrate can just swap out and attach with the enzyme. Whereas if you had a fixed number of enzymes and a fixed number of substrates, then one could either get saturated, and then the time taken for the reaction is still the same, or slower than compared to increasing the energy. So, it could be, either it could be like saturated.
Yeah. Yeah. So, well, if you increase the amount of protein, that's also the same thing as what you said, increasing the amount of the substrate. You can increase the amount of protein, but if you don't provide more substrate, then the increase will not happen very much. So you must increase everything together to be able to get something that is, to have a big increase, you must increase both together, the substrate and the enzyme. Okay, but usually substrate is it's abundant, more abundant than the enzyme. So usually it's not a problem, but if you really want a lot increase, then you must provide more substrate to more enzymes you are doing. What about, let's say you are a bacteria and or a cell, a unicellular organism is easier to understand sometimes, and you are in an environment which is very challenging. The conditions can change very much. And you must be able very quickly to adapt to your environment. Because if you can't, then you just die or you freeze or whatever. So what kind of advantage acting on the activity of the enzyme will provide compared to acting on the number of a given?
In a quickly changing environment, the enzyme could deform. That's the word. When enzymes deform, I forgot what the word is.
You may denature or something. Well, yeah, if the changes in the environment are too bad, then of course you will kill the cell. But if you imagine, what is the best regulation? What is the best way to be able to quickly adapt to the environment? Is it to change the number of enzymes, or is it to regulate the activity of the enzyme? What's the fastest control? What's the fastest control? Think about all the steps that are required to change the number of an enzyme. You must activate transcription of the gene. You must then translate the messenger RNA. Then, that takes quite a long time, right? If you want to make more of an enzyme, that takes quite a long time before the enzyme increases sufficiently to increase the reaction. Well, changing the activity of an enzyme is almost immediate, because you just need to have the right control that will directly change the activity of the enzyme. So that's much, much faster and enables the cell to actually react much more quickly. Then, how is this kind of quick control of the activity, how does it happen? Well, you can see this point here, that the catalytic activities of enzymes are often regulated by other molecules. So let's imagine a metabolic pathway in which you have substrate B here is used in a branch pathway to synthesize Z, right? And then there is something called feedback inhibitor. This enzyme that mediates this reaction, when it sees B, it will act on B and synthesize X, and then maybe a different enzyme or the same enzyme will synthesize Y and Z. But then Z can then feed back on the activity of the enzyme itself to stop. So what it means is, there is enough of that in the cell, we don't need any more. So please stop making it, because we have enough. We don't want to have too much of that. So that's called feedback inhibition, all right? But there is another term which I have not mentioned so far. This is allosteric inhibition. Allosteric inhibition means that most enzymes will have a binding site for their substrate. So that enzyme here will have a binding site for B, but they will have another binding site. Not just one, but they may have many binding sites for other things, like the product of the reaction they catalyze. And so when these products bind on the binding site, they will change the shape of the enzyme so that the reaction here will stop. Which means is that when they bind to the enzyme, the shape of the enzyme changes and cannot bind B anymore, and the reaction stops. So when Z decrease, because this reaction has stopped, then more enzymes without Z will happen, and then the reaction will start again. So this is called allosteric feedback inhibition. And it can be extremely complex. Most of the reaction in the cells, they obey that rule. Okay, so you imagine here, this is the synthesis of methionine and isoleucine from aspartate. This is the conversion of amino acid. And you can see all these reactions are mediated by different enzymes. And you can see the amount of negative reactions that are operating. So you have the enzyme here, the first enzyme that transforms aspartate into aspartyl phosphate here. You can see lysine is an allosteric inhibitor. Threonine is an allosteric inhibitor. Homoserine is an allosteric inhibitor. So very often, many products of a given reaction will be allosteric inhibitor of the reaction that produced them. And you can see methionine here is not, there is a branch pathway here. So methionine will not feed back here much, because maybe you still need to make isoleucine. You have too much methionine, but you need to make isoleucine. And if you inhibit this reaction, it will be not so good, because they both will stop being made. So instead, methionine will stop the reaction, the branch reaction that produces it. And same here, isoleucine and threonine will sort of inhibit their own reaction. So this is all allosteric feedback inhibition. That being said, for amino acid and also for nucleotides, not only feedback inhibition is important, but positive feedback is also important. There are some positive regulations as well. In this case, all what you see is negative regulation. Methionine inhibits its own synthesis, isoleucine inhibits its own synthesis, threonine, and so on and so forth. But sometimes allosteric regulation can activate enzyme instead of inhibiting. So if you look at this pathway, and if I tell you that what's important in nucleotide synthesis and amino acid synthesis is the balance between them. Having excess of one is not so good. Because if you have huge excess of one, then you can make mistakes in the incorporation of nucleotides or amino acids. So, if you look at this pathway, what would you suggest if there is a positive regulation, if there is a positive allosteric regulation? Where could it happen? Think about the balance. Balance is important. Having an excess of one is not so good. So where could be one example for positive allosteric regulation? Something that would bind an enzyme and activate its activity.
A product activate next reaction.
Like, for example, be more precise.
Small square anywhere.
Like, let's say, methionine.
No. This.
This one.
Next arrow.
This one.
Yeah.
Yeah, it could be. It could be that the abundance of substrate will promote. Yes, that's true. But then this is not allosteric. If you have more substrate, then they will occupy the binding site more. This does not involve the existence of another binding site. This is just the reaction site of that reaction. So what I am trying to see here is, imagine if there is another binding site on the protein, which is not like, for example, you take this one here, and of course there is a binding site for aspartate semialdehyde that will make homoserine, but then something else might bind to the enzyme on a different site and regulate this activity. So of course, each substrate regulates its own transformation. That's true, but that's not really called allosteric. So is there, for example, let's focus on the amino acid that we see, right? Is there any amino acid who could regulate, positively regulate another reaction than the one that produces?
Lysine activates the reaction from aspartate semialdehyde to homoserine.
For example, yes, this is one very good example that the balance between lysine and methionine isoleucine is very important, okay? And if you make a lot of lysine, not only lysine may inhibit this reaction and inhibit this reaction, but lysine may say, hey, instead of making me, make the other guys, because I'm already too much. There are too many of me. So please, may activate this reaction instead of this one. So that's a potential positive regulation here. You are right. What else? It's all about balance. What else could you add? So usually these reactions, as you correctly spotted, they will happen where there is a branch point, when you have two possibilities. So that's a very big hint. Where else could you have an allosteric positive regulation? This is kind of exam question, this kind of stuff. I give you a graph and I'm asking, you know, where could you, how could you? So that's one. That was what spotted. This one. Yeah. What else can you see? It's essentially the same, just in a different location. Think about branching point. Whenever you have a branching point, there is a danger of having unbalance in one of these branches, right? So where else you have a branching point? And where else could you have positive feedback? I mean, allosteric positive regulation.
Methionine activates
Exactly. Activate what?
homotherine to
Yeah.
isoleucine pathway, and isoleucine activates homotherine to
Absolutely.
methionine reaction.
Yes, absolutely. So they will each inhibit their own pathway via allosteric inhibition, but they will also promote the other pathways by allosteric activation. So these things are very, very possible. Allosteric enzymes have two or more binding sites that influence one another. Look at this one. It's one of the, well, it's a very well-studied enzyme. And as you can see, it contains many different subunits. This enzyme, called aspartate transcarbamylase, is an enzyme that commits reaction to the synthesis of CTP, UTP, all these nucleotides. Okay. As I told you before, it's very important to have a balance of nucleotides. You don't want to have too much of one, because otherwise you can make mistakes. This enzyme will potentially make a lot of them. But as you can see, it's on, and this is the active site of the enzyme. So the precursors of nucleotides will bind there, and this enzyme will transform them into nucleotide triphosphate or whatever. Okay? But if you have already too much of nucleotide triphosphate, it's not good. You must stop that from happening. So one example here is that CTP, which is one of the products synthesized by this enzyme. It's not direct. This enzyme does not synthesize CTP, but synthesize something that is then later synthesized into CTP. Then CTP can bind to a different site called regulatory site and inhibit the activity of these enzymes. So when CTP is abundant, you don't make more of the precursors to make more CTP. That would be wasteful. So this is one example of allosteric regulation. And allosteric, once more, means that something different from the substrate will bind to a different site of the enzyme to inhibit or to regulate, more generally, its activity. So that's one of the well-known examples. Another example is this one, and in this case it's a positive regulation. There is an enzyme that actually works on glucose. Okay? It's work on glucose to produce energy from glucose. But there are two binding sites. There is one binding site for glucose, and there is one binding site for ADP. And if there is no ADP present, then the configuration of the enzyme is not good for binding glucose, and mostly the reaction will not happen. But once ADP is present, it will bind this enzyme, the configuration of the enzyme will change, and will be able to bind glucose with a high affinity and carry on with the reaction of catalyzing glucose to get energy. As you can see here, without ADP, it's a little bit active, 10% of activity, but with ATP, the enzyme becomes 100% active. So what's the relationship? That's a very easy question. What's the relationship between ATP and glucose? What is the advantage of this reaction? Like if you remove the ADP component and you make an enzyme that always works on glucose, compared to that case, what's the advantage of that point? What's the relationship between ADP and glucose? Why would you need ADP to activate an enzyme that will burn down glucose?
Abundant ADP means shortage of ATP, so with glucose, cells should produce ATP.
Yeah, exactly. So an excess of ADP means that there is not enough ATP, so the cell must burn glucose or other sugars to get energy. So this is exactly what it means. If you don't have that, then you cannot adapt production of energy according to your need. Okay? So this is very, very critical for the cell. Okay, but simply, phosphorylation can control protein activity by causing a conformation change. We have already seen something like this. You imagine a protein that has a serine. It's usually serine. It can be threonine as well. But let's say in this case this is serine. It has a side chain that can become phosphorylated. So you can see this hydroxine here becomes phosphorylated. And this is phosphorylated by a protein kinase that will add a phosphate from ATP onto the serine. And this, once it's phosphorylated, it can then be dephosphorylated by another enzyme called the phosphatase that will remove this phosphate and will regenerate the enzyme, the original unphosphorylated enzyme. And the role of this phosphorylation can be very different. It depends on the enzyme you are studying. Some enzymes, they become activated when they are phosphorylated. Some enzymes, they become inactivated when they are phosphorylated. And most enzymes have more than one phosphorylation site. So it can be extremely complex, okay, how they are regulated. And it's not only the activity that can be regulated by phosphorylation, it can also be where they are in the cell. Do they go into the nucleus? Do they stay in the cytoplasm? Many things can be regulated by phosphorylation. Covalent modifications also control the location and interaction of protein, as I told you before just now, and this is one case of a protein called P53. P53 is a very important protein because it regulates the cell cycle, and it is regulated in not a good way in many cancer. In many cancer you have an increase of P53, of the function of P53, so it is one of the most it's very important protein, but it can go bad very frequently and cause cancer. And because it's such a key protein in the cell, it is heavily regulated. And what you can see here, it's a schematic representation of the protein from the amino side to the carboxyterminal side. And you can see all of these sites can actually be modified by different covalent modifications. You have phosphorylation sites. As I told you, there can be many for one protein. You have acetyl. So some amino acid can be acetylated. Some amino acid can be ubiquitinated. So you have many levels of regulation. This is only one example with P53. Yet other enzymes, they will depend on phosphorylation, but one phosphate will not be directly added onto this protein. In this case, these GTP binding proteins, they are a little bit different. They actually bind directly GTP. They bind GTP, and when they bind GTP, they become active. But GTP that is bound to this enzyme can be hydrolyzed into GDP that is still bound to the enzyme. But when this enzyme is bound to GDP, not GTP, this enzyme becomes inactive. And slowly, GDP, because the affinity of binding of GTP is lower than the affinity of binding GTP, then slowly GDP will go out of this enzyme. This enzyme, still off, will be bound with nothing, and the binding site for GDP will be replaced with GDP. And this is very fast, and the enzyme will recover its activity. And you can imagine that this is also can be regulated. How quickly you exchange GDP for GTP can be regulated. So you can regulate the cycle of activation, inactivation. It's another type of regulation. It's called GTP binding protein. So it's still something to do with phosphate, but it's different from a phosphate that is added by a kinase like this. It's completely different. And probably one of the best examples of how big can be the effect of only one ATP or a series of ATP are the motor protein. And this is, I will show you a great movie later on. But imagine this is, for example, actin filament or microtubule. It's a kind of chain in the cell that's something like a protein can work on. Okay, it's like a rope. And you imagine that protein has three configurations. This configuration, and this configuration. Okay, three. And if you allow this protein to, at will, at random, shift from one configuration to the other, what will happen? So you can see this is stepping, right? This is 1, 2, 3. 3 stage. If you allow the protein to randomly shift between each of the three, will the protein go forward or will the protein go backward? On this blue road here.
On the same place.
It will stay on the same place. Randomly, it can go forward every now and then, but it will go backward as often. So actually it will stay in the same place because all of these are perfectly reversible. So it's completely random walk. The protein will go nowhere. However, if you actually make one step dependent upon ATP binding, ATP hydrolysis, and release of ATP, then this becomes directional. This protein now, instead of going forward, will always go backward because the reactions are not reversible. One binds ATP, the other one hydrolyzes ATP, and then ADP plus phosphate are released, and the new cycle of ATP binding can happen. So this protein will only go forward. This looks very stupid. It's a very stupid drawing, but look at this one here. I don't like the fact that the background is completely white, so I'm going to make it completely black. This is one picture I took in the laboratory here. This is a cell taken from the brain. This is a cell taken from the brain of a mouse. It's not a neuron. It's a cell called an astrocyte, which is a big cell that feeds neuron. And I labeled mitochondria with a green fluorescent protein. This is not green. This is monocolor microscope. But each of the dots that you can see and this very bright thing is the nucleus, but it's surrounded by mitochondria. Okay. And if I actually play the film, if the film wants to be played, great. I love Microsoft PowerPoint. I love Microsoft. So I'm going maybe to play just the film by itself. So you can see there are mitochondria who actually go towards the nucleus, and there are mitochondria who are going away from the nucleus. Like for example, this one, it doesn't go anywhere. Sort of stays there, but it went. And if you see some really point like this, they can really move. And this is really mitochondria moving on these microtubules or actin cytoskeleton. So this is application of what I just told you. This molecular motor allows cell to actually carry its content, like mitochondria. And if you want to see at the molecular level what happens here, then there is a great film which we can play. Cytokinesin work. That's this guy here, and this is much better. This guy has pumped on YouTube this. That's really a great film. That's sort of, it's a synthesis of everything I told you just now and previously. So you can see, for example, in the membrane, all the protein that stick out and allow cells to interact with membrane. This is also, what do you think this is? What do you think this is? From what we have seen. This is the cell membrane that is completely filled with protein that are floating on the cell membrane, and that's underneath of the cell membrane. It's all the proteins of the cytoskeleton that interact with the plasma membrane. And we are going inside the cell where we can see all the cytoskeleton protein, like, for example, actin. This is the polymerization of actin. The next we are going to see is protein that interacts with actin to cut actin when it's not needed. This is a microtubule, or you can see each protein subunit assembling into the microtubules or being disassembled when it's not needed. And this is what I just told you. This is a protein walk, where this protein is called kinesin, and it's walking on a microtubule. And it's walking because the movement is mediated by the binding and the hydrolysis of ATP. So this is exactly what happens there. So I will play this one again because I find it's very cool. So what you have there is a mitochondria who is being transported by kinesin walking along the microtubule. And it's all due to conformation changes because of ATP binding. So anyway. Of course it went down. All right. Well, as you can see from the film we just saw, some groups of protein, they will assemble into huge protein machine, okay, for a specific purpose. And in the book, this is the case you have, where you have 3 proteins, and they can interact together for a very specific purpose. In this case, it's a little bit silly. This is the opening of a safe. But if we have time this year, we will see how this example is almost exactly what happens for the synthesis of DNA. For the synthesis of DNA, there are so many proteins that interact with each other, and they communicate with each other to be able to actually synthesize DNA from the double helix, which actually involves a lot of different reactions that all need to be synchronized. Okay, so what we are going to see now is how can we analyze protein. All the things I've told you, they were understood from experiment. But experiments very often require you to extract protein from whatever samples you have, from cells, from liver, whatever. So the first step to analyze protein is very often to break cells and tissue. And you can break cells in different ways. One of the most powerful ways you have to break cells is to use ultrasound. You have your sample that contains cells, and you fire ultrasound into your sample. This will actually break the cells into its component. But of course, you can also use detergent in the same time to promote the disassembly of protein. Something that you can do is also forcing the cells through a very small hole. For example, if you use a syringe, you can also homogenize cell and liberate their component by forcing cells into a syringe. Or you can shear cells by using a pestle and a mortar, the equivalent. Okay? Where what happens actually is that the cells will be forced in a small space between this pestle and the tube there. So the principle is very similar to that one. And at the end of this homogenization, you have cells completely broken up. And you can see you have 3 mitochondria and 3 nuclei. These can be separately purified. If you're only interested in nuclei, you can just simply take the nuclei. If you're interested in mitochondria, you can simply take mitochondria. How are you going to do that? How are you going to do that? You have broken up the cell. And the way you have broken up, they are very different ways. It's not as simple as what I just told you. But let's say what you have will not break the nuclei, will not break the mitochondria, but will break the cell. how could you only get nuclei? You know already. How do you only get nuclei from that? You have a mix of different stuff. Actually, if you just take a look at the picture, you can see you have a mix of particles of different sizes. And that's the key. Particles of different size mean particles of different density, probably, different weight. So how can you differentially purify them? By centrifugation, simply. If you centrifuge with specific settings, not so fast, then the heaviest thing will go down. If the heaviest thing that you have here is the nuclei, then with very low centrifugation, you will have the nuclei falling down your tube because of centrifugation. And the mitochondria, microsome, and all these things will stay in the solution. So that's one way to purify these things. And if you want mitochondria, you see they are a little bit smaller than nuclei, so you can first centrifuge, low speed, so you get the nuclei, you get rid of the nuclei, then you centrifuge a little bit faster, and you get the mitochondria. This is the principle of differential centrifugation. But there is a different way by which you can separate protein, because proteins, they have different size, shape, charge, hydrophobicity, and so on and so forth. And all of these characteristics can be used to separate protein. This is what we just saw now. By centrifugation, you can pellet only specific protein. Bigger protein, larger and more dense component will go into the bottom of the tube, and the smaller and less dense components will stay up. That's one way. Differential centrifugation, this is what I just told you. So first you pellet, for example, the nuclei, then you get the mitochondria, then you get different fractions. That's another type of centrifugation that actually involves having a gradient of sucrose. So this is low concentration, this is heavily concentrated, so there will be a gradient of density, which means that after centrifugation, each cellular component will stabilize at the location of the gradient that corresponds to their own density. And this is called velocity sedimentation. You have equilibrium sedimentation, which is a little bit similar, but it's based on the buoyancy of these proteins. And column chromatography, we have seen something similar already, where you have a column with a matrix inside, and depending on the nature of your column, you will have different constituents of the cell. They will be separated, and you can collect different fractions. So in this case, for example, if red is a little bit bigger, it will go first, but it could be the other way around. It could be that because red is a little bit smaller, it will go faster. So it can be really different according to the column that you have. And these are the three different columns that you can have. So you have this ion exchange chromatography will separate protein according to their charge, because you have positively charged beads that will retain negatively charged protein. So the more negatively charged a protein is, the more retained it will become. And if you have neutral components or not so negative, they will actually go very fast through the column. You have this gel filtration chromatography, where the beads here have pores. Inside the beads, you have holes in which cellular components can enter. Okay? Of course, you can buy beads with pores of different size. You know what you want, so you can buy whatever size of pore you want. And in this case, the big protein that cannot enter into the pore will actually quickly go at the bottom of your column. But the small protein who can enter into the pores of your beads very slowly will move. Why? Why the big protein will quickly fall at the bottom, while the small protein will remain? Can you easily explain why this happens?
Small proteins are stuck by the bees.
Yeah, small protein will be stuck by the beads, but essentially because they can enter into the beads and the path is not straight, so the distance they actually have to make is bigger than big protein. So this is what will happen. But you can also specifically purify protein by affinity. So in this case, for example, you have beads, as we have seen earlier. We have beads that maybe have antibodies, or maybe they have substrate bound to these beads. So you will retain protein that interact with whatever coats your beads, and this is called affinity chromatography. Okay. So these are the three different types of chromatography. And what is great, it's something that I rely on quite a lot. I mean, it's a very simple textbook, but it actually contains things that most of the scientists are using almost daily. So I have a student who almost do that every day. So you have, these beads contain a protein or whatever, and you want to know, I wonder what interacts with this protein. It's a new protein, we don't know its function. So to understand the function, what kind of protein does it bind to? So then if you have this affinity column, then you can bind this column to this protein, and then you put a mixture of protein from a cell, and then proteins that interact with this one will actually bind to it, and the proteins that do not interact will actually be washed out. And then if you add a buffer that is a little bit more stringent, that maybe contains a bit more detergent, then you can actually get the protein that interacts with this protein. So this is a variation of this affinity chromatography. So not only you get protein that directly interact with whatever you want, but what also will you get? In this case, it's nice. This blue, and this red, they directly interact with this green one. But what will you get as well? That you actually don't really know which case will be which when you see what protein you have. So here we can see all these proteins. This is the case where they directly interact with green. And at the end of your experiment, they say, all right, these are X-binding protein. Yeah, that's true. But that's not everything you will have. What else will you have that can make your analysis a little bit more complex? What else will you have in your final purified sample? What else will you have? That's easy. If they do not directly interact with your green protein here, what could they do? Yeah, these proteins also will happen. Yeah, you can wash them out because they don't interact with anything. But what else could you have in your final element, in your final mix of so-called X-interacting protein? What else will you have? What else will you have? What else will you have?
Salt or base or acid.
Yeah, of course, you will get, the components of your buffer you will also get. You will not only get protein, you will also get your solvent, yes. Your elution buffer you will get. But you may get protein which actually they may look like they are interesting, but they may not be actually very interesting.
Protein that binds to
Yeah, continue. Protein that binds to protein.
that bind to protein X binding protein.
Yeah, exactly. That's exactly the case. So not only you will get protein that interacts directly with green, but you will get protein that interact with protein that interact with protein that interact with protein that interact with protein that interacts with green. So you have a mix of protein that binds stuff, and the thing that you know is that somehow they are associated into the complex there. It's great, but do they really bind directly to green? You don't know. And this can make things complicated. So, imagine you do this experiment and you are interested in red here. You like red, but you don't know if it really interacts directly to your protein. So what is the next experiment? In this case, you use the mixture of protein. So what you did, typically, you got cells, you mashed them up, like that. You made sure that all these were broken up and all the proteins were released. You add them onto your affinity chromatography column. You get these proteins, and the next step is you want to see if they really directly interact with your protein. What do you do? What do you do?
Do the same experiment only with red protein.
That's true. So you do an experiment where you don't have a mix of different proteins. You only have your column bound to protein X, and onto the column you add a purification of your red protein. Only red protein. And if it's still retained by the column, then you know you've got a direct binding. Okay. So there are different techniques that you can use to analyze protein, and one of the most commonly used is this gel electrophoresis, as I explained a little bit before. In this case, what you have here is a gel. Usually for protein, it's a gel made of acrylamide. Acrylamide is very long protein that can actually cross-link. So it makes like a network of long chain. Okay. And you have onto the gel here, so you have your gel, and on top of the gel you have little reservoir, you have little holes in which you can put your sample. And then you have a cathode, you have an anode, and then when you apply current, your proteins will be carried by the current down. And usually, the faster they go to the bottom, the smaller they will be. And of course, you must use detergent like sodium dodecyl sulfate, this very big ionic detergent, very powerful. This detergent will completely denature your protein. Your protein, because of SDS, will be completely negatively charged. Completely. So what happens is that what will actually happen in this case? Why do we do that? Yes, we do that to solubilize the proteins, but we also do that for a different way, for a different purpose. And it is related to what I said just maybe 5 minutes ago, maybe 2 minutes ago. So we use SDS to denature the protein and to coat the protein completely with SDS, which all protein will become negatively charged. So what's the consequence? If you don't do that, what happens?
Proteins are usually charged, so without SDS, the result will be affected by the charge, original charge.
Yeah.
So precise to result will be precise with molecular weight.
Yes, exactly. That's exactly, no, you said it very well. So proteins each protein has a different charge, but you don't want charge of protein to be an issue. You just want to separate protein according to their size. So therefore, this is why you use SDS. And then in this case, charge is not an issue, and you know that if a protein goes quickly to the bottom, it's because it's smaller than the protein that remains up there. But you have some problems, for example, as you know, you may have some disulfide bond between proteins. So you must use as well some reducing reagents to break these bond, so that you have single peptide running into your gel. So it's a very common technique. But if you want not only to separate protein according to their size, but you want also to separate protein according to their charge, then you can do a two-way. You can first separate protein according to their charge, and then separate protein according to their size. So this is called the 2D electrophoresis. So the first, you establish a gradient of pH, and each protein will equilibrate at the region that corresponds to its isoelectric point, okay? Where the protein is not negative, not positive. And then once you do that, then you put this strip of protein all organized across their charge, and then you put this strip on top of a gel like that, and you separate protein according to the charge, to their size. And what you get, instead of having something like that, you have something like that, which is two-dimensional. This is the size, and here it's the separation of protein according to their charge. So this is the first separation you have done based on the charge of the protein, and then this is the second separation that you have done according to the size of the protein. So now it's very clear because you have each spot here corresponds to a given protein with a specific charge and a specific weight. And what is great with these things is that you can actually punch out proteins and you can analyze what they are. You can break up the peptides and you can sequence each of these peptides to actually identify what is this protein there, for example. This is kind of a useful technique. It's difficult to do well, but it's very useful. And You can see in this table 4.2, it's different historical landmarks in understanding of protein. I'm not going to ask you at the exam what happened in 1934. I'm not going to ask. This is for your information, okay? This is not part of the exam, but it's kind of useful, useful knowledge. Wow, it's kind of interesting. So, see, for example, the name protein was coined in 1838. Not that long ago, if you think about it. Okay. And then that's the end of the chapter, and I think I will end up there. So, you said that the second point, once you get proteins that interact with your protein of interest, you want to confirm whether this protein really directly bind. So the only thing that you need to do is to only put red protein. Just remember, how do you do? How do you do? How do you make this red protein? You must have a mix that is composed you must have a solution that is composed only of this red protein. How do you do? How do you do that? That's a hint. That's very big scale. So you have a protein of interest that you think interacts with X. So you want to confirm that it really interacts with X. So not only you must have X, but you must have your protein that interacts with it. So how are you going to make these proteins? You cannot use a mix of proteins. It's a little bit dirty. Of course, you could use an antibody against your protein and purify each using affinity chromatography, using your antibodies. That's one way to do it. But usually, it's not so efficient. No, it's better to actually produce directly the protein that you want. How would you do it? If you want a big amount, and indeed, imagine you are a pharmaceutical company and you want to make a lot of insulin. Insulin is a peptide. How are you going to make a huge quantity of insulin that is used for treating diabetes? How are you going to make a high amount of protein? How could you do? You can go, and anybody is okay. You can jump. Yeah, you want to say something, I see. You have something burning. Go for it.
Yeah. I've never seen insulin made in human body. Which organ? Which organ? Which organ is?
Thank you, it's a good question. Which organ? Which organ makes insulin in your body? Where is insulin made? Where is insulin made? You guys should know. Where is insulin made? Insulin どこにある？日本語でも OK。Insulin. 肝臓?
Pancreas.
Yes, 膵臓. It's the pancreas. So you have different cells in the pancreas. One will synthesize insulin, the other one will synthesize glucagon, and they will sort of balance each other. So yes. So actually, originally, if you want to make a lot of insulin, you kill a huge amount of cows. The insulin will work on your receptor of insulin. So that's okay. But you must kill a lot of cows. So it's okay, it's right, but there is a much more efficient way. It's kind of expensive because you must have a lot of cows. Now, the only thing you must have is much smaller than a cow. And you cannot have billions of cows, but you can have billions of that. Yeah?
Using mouse.
You use mouse, yes. You could also use mouse, and it's smaller than a cow, but still it will not be very much efficient. And then imagine maybe to have, I don't know, 10 milligram of insulin, then maybe you would need to kill 1,000 mice or something. So it's a little bit costly. It costs a lot. And it's not so good because people will sort of bomb your car because you're killing too many animals. So not mice. But you are right. You have to go down the ladder of living things. So you make something produce your protein, whether insulin or whatever.
Cells.
Cells, yes. What cells? You can use human cells, or you can use mouse cells. Just not the mouse, but the cells of the mouse. And then you can produce protein. How do you produce protein? How do you have to hijack, or you cannot just ask the cell, say, please, can you make insulin? Right? The cell is not going to do that if you just ask. So you must hijack the protein synthesis of the cell. Huh? Virus is great, it's a great idea. But viruses will not produce anything by themselves. They need cells. You can use virus to hijack the system of the cell. Yes, that's true. So you can use viruses that will inject into the cell a small bit of DNA that codes for insulin. That's one way you can do. But that again, it's a little bit costly, and mammalian cell requires a lot of maintenance. They are usually limited in how fast they grow. So it's a good idea, but you can do better. If you really want to have a lot, then, and cultivating mammalian cells, it's very expensive because you need to have amino acid, you must have protein, you must have vitamins, you must have serum because cells do not grow well without serum. So you also have to kill cows because you need serum to grow your cells. This is not so good. So what else? What's remaining? What's remaining? You cannot use mice, you cannot use cows, you cannot use mammalian cells. So what's remaining? If you go down to the simplest living thing. Yeah, I heard something.
You said it first.
Yeah, I heard something.
Bacteria.
Yeah, bacteria. Nowadays, most of the proteins are made in bacteria. If you want to have a huge amount of protein, you use bacteria. What you have here, they are these reactors that contain bacteria to actually make all these proteins. So this is what people use. So what you need to do is, you don't need to use viruses, but you just hijack the protein synthesis system of the bacteria to produce a given protein. And then you can grow these bacteria, they will multiply, and they will produce your protein of interest. And what is great is that if you always give medium, which is very easy, this is very cheap, they will grow happily, they will multiply, and you can ensure a steady supply of proteins like insulin, for example, for diabetic patients. And that's the end of the chapter. Any questions about protein? Remember, next week we have practice test. And it's only 10 minutes. Maybe I can finish chapter 5 in 10 minutes. Is it a good idea? No? Okay, I will not finish chapter 5. Chapter 5, it's about DNA. It's a very short chapter.
And next week test until the end.
Next week, it's until now. Next week test, it's until now. Until the end of this chapter. Okay. Do you have any question first? This test is going to. I don't know yet if it's going to count or not. It depends how good you are. If you are very good, it will count. If you are all bad, it will not count. So you have nothing to lose.
So no essay questions where you need to remember all the terms.
No! There may be maybe 10% of essay questions. But what we are going to do next week, it's not a test of 1.5 hours. It will be a test of 45 minutes or 1 hour, and then we are going to review the questions together. Okay, so this is just a practice. But if you are doing well, it may count for the final score. Usually, the final exam contains more essay type questions. But when I say essay type question, like you have two types of question. Which proposition is correct? A, B, C, D. Which is correct? That's easy. But there are different type of questions. Your friend come to talk to you and he discuss about the possibility to synthesize a protein. And he has a way, but he doesn't know if it's going to work or not, and he's asking you advice. What would you say to him? This kind of questions, where you have to explain a little bit more.
Final test?
No, for next week, you will have some of these questions. But the final test usually contains a bit more of these more complicated questions. Maybe 10% or something, 20% or something. Twice the amount. Because these are the questions that are interesting. But vocabulary is not usually a big issue. Vocabulary is not a big issue.
Books or slides are permitted?
I'm not sure. I think it actually didn't make. I think last year I accepted textbook, but the distribution of marks was just exactly the same as the year before without the books. So yeah, okay. Let's go with the book. For the final exam, book will be allowed. But the questions will be more difficult. But the book that you can bring is only English, okay? English book. English book. Because it's kind of E2 lecture, so we have to.
Next week, no book.
That's right. Next week, no book. And you'll see how hard it is. And if you think it's too hard, then we sort of can easy. I haven't made the final decision on whether you can take it.
It's a bit of expensive book, so like PDF. It's PDF fine.
Yeah.
PDF. Buying the book.
PDF is fine. Yeah, I mean, it's also quite heavy to transport. Yeah. Okay, so that's the end of today's lecture. If you have a question, please ask. And next week we're going to have final test. No, a bit. See you. Bye bye.
テストの持ち込みって何なんか期末テストは英語の本を持ち込んでいい。来週のテストは何も持ち込まない。
はい。
ディクショナリー。ディクショナリー。
ディクショナリーもちろんいいですよ。これはなんかね、辞書とかそれは全然大丈夫。全然大丈夫。けどテキストブックはダメですよ。テキストブックダメです。いや、ディクショナリーは全然。大丈夫？
They probably saying final test with PDF is okay.
No, I don't think so. Final test with PDF is okay. Yeah, it's okay. PDF is fine. If you have the book as a PDF, that's also okay.
Internet with device with internet access.
Yeah. So that's okay. The questions are not only intended. The questions are not only intended. But I'll keep an eye on CE. So don't need to.
持ち込みなしの方が簡単なんじゃないかな。
But I think the book probably don't need the book. But the questions that I am asking, they are questions you need to think. They are not questions that you can find in the book the answer. So you can bring the book or not, it actually doesn't matter. And actually, my advice is not to bring the book. Because if you bring the book, and I see because I have looked at students, if you bring the book, you get a question. Instead of thinking, you look in the book to look at the answer. So that's actually not so good. So that's just advice. That's an advice. Just think about it. Use the dictionary to understand the question in Japanese, and then maybe make the answer in Japanese, and with the dictionary translate in English. This is what I'm doing. Bringing the book, it's not helping.
So prohibit the book and make question more easy.
Let me think about it. We will see the level. We will see the level next week. It's not that difficult. 痛いそう。大丈夫よ。大丈夫だ。仕事に。Remind me, you are first year?
Yes.
That's not possible. You are lying. You are really first year. You are really first year.
Really first year, but I was in another university to study computer science.
全然関係ない。So you have first done computer science for how many years? 2, 3 years? 1 year?
5 years.
Oh, so you are graduated already?
No.
So
Dropped out.
you dropped out. なんか面白くなかった。So you done computing science and then you dropped out and you started now here.
Yes.
Really? But
I not officially take the biology or medical class.
when did you do that?
Previous university.
When you were doing computing science, you also not officially took biology. So yeah, your level is pretty high. It's pretty. I'm very, very surprised. I'm very, very surprised. So that's very nice. That's very nice. What do you mean non-officially?
In Japanese, moguri.
So you went to the lecture, but you did not get credit or anything.
Yes.
Just for the lecture. Interesting.
And in studying for entrance exam, I choose biology
I see. Interesting.
and study biology in Japanese.
I see. So this is why you have very high level, quite high level. But I am impressed because it is very rare
Rare.
to meet somebody like you who is different, doing different things,
Doing
right?
different things.
Yeah, I think it's kind of rare. It's good. I mean, it's good, but it's very rare because usually in Japan, I notice most of the students, they do the same thing at exactly the same time.
But I'm a medical student. It's in my specialized
Yeah,
area.
but it's, first you wanted to do computing science, and then, I mean, I think it's good. It's pretty good. Sometimes you need to find, my sister also was like this. My sister, she started university in history. And then she decided to change, and she stopped history and she started philosophy. So it was also a bit similar, I suppose. She dropped out. She just think it wasn't very interesting.
Was philosophy interesting?
Philosophy?
She feels philosophy is interesting.
Yeah, she liked philosophy. I don't think it's very interesting, but she liked it. She liked philosophy. And and my wife also is a little bit not conventional person because she went to international university in Japan, and then she went to the Philippines, and she became United Nations worker. But then she started a PhD. So she first worked, and then after maybe 10 or 20 years, 10 years or something, she started a PhD.
For 10 years
she
in
worked.
United Nations.
Yeah, about maybe 5-10 years, United Nations. She went to Philippines, Rome.
It's very high career.
Yeah. So she had a Mitsubishi grant when she was in Japan. So
Grant.
I mean, she was a recipient of the Mitsubishi when she was at university. So Matsushita, not Mitsubishi, Matsushita. Matsushita fellow. But she is not at all conventional. She did many different things. So, in Europe, or in Japan, many people do PhD. Graduation, master, PhD, right? So what do you want to do? You want to be physician? You want to become physician? Or what do you hope to become?
Researcher on medical equipment or something like I now think I want to produce medical equipment which can diagnose psychiatric disorder.
You said so in the beginning of the lecture. You're interested in medical equipment for diagnostic. That's interesting. So for that, maybe physics or informatics also might be important.
Yeah. And medical science.
Yeah, engineering, medical science,
and
大変。大変。